Hello, everyone, and welcome to another Genetic Engineering and Biotechnology News webinar. Our presentation today is entitled Multiple Attribute Monitoring of Biopharmaceuticals Using Mass Detection. I'm Jeff Bogaliskis, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. The development of biopharmaceutical compounds requires vigilant monitoring of formulation and production reagents for both safety and accuracy. While most current production procedures involve the detection of analytes through the use of optical detection methods, industry trends and increased regulatory stringency has manufacturers incorporating mass spectrometry analysis more frequently, due in large part to its ability to analyze more complex molecules within a shorter time frame. Advances in technology and measurement sensitivity have allowed for the development of equipment that employs the use of both UV and mass data in implementing multiple attribute method analysis that typically targets downstream processes, but are increasingly being expanded to QC assay protocols. The demand for cost-effective and user-friendly mass detectors for routine monitoring of multiple biotherapeutic attributes is on the rise. Let's meet our speaker for today's webinar, who will discuss the implementation of multiple attribute methods assays and how they can aid production analysis. Sean McCarthy is a Senior Manager for the Waters Corporation. Dr. McCarthy will provide data from application examples which show that combining UV and mass detection delivers statistically independent data points that can enable the direct and efficient implementation of multiple attribute monitoring assays utilizing the Waters AcuityQDA detector. Before Sean begins his presentation, I want to encourage you to submit questions for our Q&A session at the end of the presentation, and we'll try to answer as many questions as we can. Simply type your question into the Q&A box on the left-hand side of the screen and hit submit. So, with all the particulars out of the way, let's get our webinar started. Sean, the floor is yours.
Thank you, Jeff. Thank you for that nice introduction. As Jeff mentioned, I want to speak today about the use of the AcuityQDA for doing multi-attribute monitoring of biopharmaceuticals and how we see the implementation across the industry of using mass detection for biopharmaceutical analysis, particularly routine analysis. So at the outset, an overview of the presentation, I want to begin by discussing what multi-attribute monitoring is, and then transition into a discussion of mass detection and what the AcuityQDA is. Then move into the heart of the presentation, where I'll focus on a variety of applications that we've seen the QDA both in our lab and in our customers' labs being applied for, and then provide a brief summary at the end. So to begin, the concept of the multi-attribute methodology is something that Amgen coined the phrase several years ago. And it's something that we've seen within the biopharmaceutical industry as a growing trend and a desire across a wide number of companies working in biopharmaceuticals. The drive to this is that by implementing a multi-attribute methodology, it allows biopharmaceutical firms to improve their productivity and also the certainty in the biotherapeutic molecule that they're producing. And this is really enabled them to reduce the number of assays that would be required in terms of assessing the critical quality attributes by being able to directly monitor each of those attributes in a single method, and then offers the potential for real-time analysis and the opportunity to implement a real, true QBD approach, particularly for the process development. In addition, in terms of specification setting and routine QC analysis, that offers the ability to have increased compliance in monitoring each of those assays and looking specifically at particular critical quality attributes. And what we're seeing, and as we'll discuss throughout the presentation, in the past there have been some barriers to implementing mass spectrometry for this, largely in the cost and the complexity of the instrument. And some of that can lead into the prevention of scalability of that platform across the organization. It could lead to some issues in terms of redundancy. We then look at the AcuityQDA at a very high level. Waters, as one of our corporate missions, is to be an innovator, both on the technology advancements, so that would be introducing technological advancements in mass spectrometry, and then also over the last few years, we've really been driving to enable a larger number of people to have access to that technology. And that's really a good intersection between the access to mass spectrometry and the implementation of mass detection, which is sort of an arm of mass spectrometry. And the focus of this is to expand the access to mass spec information to a broader range of users. So back in October of 2013, we introduced the AcuityQDA as a really revolutionary product. The design of the instrument was really focused on ease of use and accessibility for a wide range of users. Primarily, the focus was on access by chromatographers who may not traditionally have had experience with mass spectrometry. And what we've seen is that the implementation of the QDA is allowing analytical chemists across an organization to generate mass information for all of their analysis. The instrument, as you'll see in a moment, is very compact, and it also is quite affordable. And it can be interfaced with a wide range of applications, and we'll walk through those throughout the presentation. And lastly, and perhaps importantly, the instrument is designed to seamlessly integrate with your existing workflows, whether they be in Empower or alternatively in MassLink, for HPLC and UPLC-based analysis. So if we look now at implementing the AcuityQDA, you can see we have what would traditionally be an existing optical-only based instrument configuration. And then moving over, when you can see the AcuityQDA circled on the right-hand side at the bottom of the stack. So it's roughly the same size as an optical detector. As I mentioned, it fully integrates with into Empower. And what we found is that there's really minimal training needed in order to use or collect meaningful data with the QDA. It really is as simple to use as an optical detector. The qualification exists, so this can be implemented within qualified environments. And perhaps importantly for many of those within the Americas and other geographies, the instrument can work on either 110 or 220 volt power feeds, meaning there's no need for additional electrical work within the lab in order to implement the technology. And as I'll discuss in a few moments, the maintenance of the instrument is also quite minimal. If we look now at the QDA, this is shown on the bottom is the graphical display from our instrument console where you can interact with the instrument. Is a depiction of the layout of the QDA. In the design of the instrument, we really designed it to, as I mentioned, to be very accessible for a wide number of users. So in that, we've created or enabled the ability to do an automated resolution and calibration check, and that's at each startup, and that's to ensure that the instrument is performing properly. And the calibrant and solutions used to do this are all contained within the AcuityQDA. In addition, the electrospray interface was optimized, and that was optimized to enable very good sensitivity for a wide range of applications with a minimum of tuning necessary, which has often been a burden and a challenge for implementing MS in downstream laboratories. If we look more closely at the graphical display, a key point I want to make here is that while we've designed the QDA to be very accessible to a wide range of users, that does not mean that the technology within the instrument is simple. So you can see in the center portion of the instrument, we have a small step-wave device, which is the technology we first introduced on our higher-end mass spec instruments. And the introduction of the step-wave within the QDA allows us to access a great level of sensitivity for an instrument of this compact size, and also lends to the robustness of the instrument for doing routine assay. We then look at the interaction with the instrument in terms of programming it. We see that it's very similar to what you would do today for an optical detector. So the Empower graphical user interface is virtually identical to that for what you would have for a PDA in terms of setting up the method. And that lends itself to requiring very little training and allows it to be added to existing workflows that may be in use in laboratories today. So instead of on a PDA where you would set a starting and an ending wavelength, in this case you would set a starting and an ending mass range for your analysis. So again, a very familiar interface, and it's seamlessly integrated with the other components that you would use during that separation, whether they be the pump or the auto sampler, all within one inlet method. In addition, I had mentioned the maintenance of the instrument. there really are two different components that would need to be serviced. One is the sample aperture. The sample aperture, if you're familiar with mass spectrometry in the past, is analogous to the sample cone. The difference with the aperture in this case is that it's designed to be disposable. So it's priced as a consumable, and it is meant to be disposed of if at some point it becomes partially obstructed. And there are self-diagnostics within the instrument to allow you to understand that you may need to service the aperture disc on the instrument. In addition, the capillary, which would be effectively the source or the emitter of the MF, is pre-assembled and there's no cutting required. So changing the capillary on the instrument can take roughly on the order of probably a minute, potentially 2 minutes to complete that process. And since there's no requirement for cutting, you're sure that you're going to be getting the same results over and over throughout each of your analyses. So in summary, we've seen that the AcuityQDA really is an enabling tool that's allowing analytical chemists to collect mass data and have easy access to mass information for all of their analyses. And that, it's doing that by delivering additional orthogonal information for the components within your analyses. And lastly, the design of the instrument makes it very easy to deploy, simple to maintain, and given its compact nature, easily fit within existing workflows. So with that, I want to transition and start to discuss some of the specific applications where we've seen the QDA be useful. First, starting with looking at peptide analysis. So this is an example of some of the early work we had done with the AcuityQDA to investigate how applicable it would be for peptide analysis. So this is an example of a standard peptide mix and looking at the different components within that mixture. And you can see that we have a very robust signal for each of the peptides contained within that sample. And in addition, highlighted within the green box are all of the ions detected for each of the peptides that were in that mixture. So for each of the peptides, multiply charged species were found for each of the peptides. If we look at the largest molecular weight peptide within this mixture, which is roughly a 3,000 Dalton melacin, you'd see that we observe 3 different charge states, and we get a very clean spectrum from the QDA for that peptide. Understanding that in the majority of laboratories, a peptide mixture is probably not the normal type of analysis, we investigated the application for something more complicated, in this case, a tryptic digest of the monoclonal antibody trastuzumab. What we found was across the entire chromatogram, we were able to detect all of the peptides that we would expect to find. And more interestingly, if we look at a separation within TFA, which is the top chromatogram, and then compare that to a separation done in formic acid, while there is a selectivity difference between these two separations, what's notable is that we're able to see all of the peptides in each analysis, regardless of whether we use TFA or formic acid. And that highlights the point I had made earlier, where the QDA could seamlessly fit into existing workflows. We recognize that the majority of routine methods primarily use TFA. So if we look more closely at that in the comparison between TFA and formic acid, shown here is a map of the heavy chain peptides, both the average mass for each of the peptides that on the left-hand y-axis, and then across the top, the various charge states for each of those peptides. And then we've color-coded whether you can detect those peptides within TFA or formic acid or in both. You can see across the range for each of the peptides, you can easily see each of those peptides within the heavy chain. And similarly with the light chain. So really the key takeaway is that we get high sequence coverage as well as important information for each of the peptides with the monoclonal antibody, and we can do this both with TFA and formic acid-based separation. And in most cases, we see multiple charge states for each peptide. If we look more closely at the types of peptides we're detecting, shown on this slide is the spectrum for the largest peptide from a monoclonal antibody digest. So this is a peptide from the conserved region. So this would be a common peptide across the majority of monoclonal antibody And what you can see for this peptide, which is roughly 7,000 Daltons, we can detect 6 different charge states for that peptide. And this highlights the fact that the AcuityQDA, even with its mass range, which maxes out at around 1,250 Daltons, for peptides, they form multiply charged species, which fit very nicely with the mass range of the QDA. We then look at the mass measurement accuracy across the range of the different peptides. The specification for the QDA is plus or minus 0.2 Daltons. And we can see that all of the peptides that we detect fall well within that range, with the majority of them being less than 0.15 Daltons, given what the expected mass for the peptide is in each of the charge states. So again, very good mass measurement accuracy. Now, if we look at some specific applications, first looking at a specificity, in this case we have a partially co-eluting pair, T26 and a T10 peptide. In this case, by using the QDA, we can very easily decouple each of those species by generating extracted ion chromatograms for each of the species, and determining their relative peak area by integrating each of those extracted ion chromatograms. This gives us the specificity to look at each of the species independently and also determine whether or not a peak is pure. Another application in which we've seen a lot of interest is in doing some quantification and relative quantification of peptides across a peptide. So in this case, we're looking at a particular peptide, in this case an oxidized peptide, at various different concentrations within the sample. So we purchased the authentic oxidized peptide and then spiked it in to a sample. And what we see is we get a very linear response for the TUV down to a certain concentration level, where then if we go to the total ion chromatogram and look at the information coming from the QDA, we can extend that the sensitivity to a lower range. And then with the capabilities of the AcuityQDA being able to do both full scan as well as selected ion recording, or SIR, we can extend that range even further. So we can extend the range at which we can see impurities across the assay quite easily. Then apply that type of approach to looking at something like an oxidized peptide. So shown in this slide is the TUV chromatogram for the T21 oxidized peptide as well as the native peptide. What we can see is that for the native peptide versus the oxidized in the TUV, there's no real discernible oxidized or peak corresponding to the oxidized species seen in the optical trace. If we look at the total ion chromatogram, there appears to be a peak there, as well as for the native as well as the oxidized species. We can get a much clearer example of on the selected ion recording chromatogram for both the oxidized and the native species in order to do relative quantification of each of those. A very targeted approach to look at one specific critical quality attribute within this peptide map. Another application in which we've seen a lot of interest has been in looking at the CDR peptides, or the complementarity determining region peptides for the monoclonal antibody. And these are specific for different monoclonals. And in this case, we've done a separation and look at both the total ion chromatogram and then generated an extracted ion chromatogram for each of the peptides that correspond to the CDR region in that antibody. And we can very easily identify where those peptides are and quickly assess an identity within that monoclonal in that analysis to assess whether or not the antibody in hand is in fact the expected molecule. So it's a very facile way to do the identity test. In addition, we've seen and heard a lot of interest in looking at deamidation. So this is an example of looking at a deamidated peptide from the CDR region, where we could see in the total ion chromatogram we have a number of species, some of which are partially co-eluting, whether with related species or unrelated species. And then we can generate extracted ion chromatograms for each of the peptides within that region. And then looking at the T34 peptide versus the T34 star peptide, which is the deamidated species. And then looking at the extracted ion chromatogram, we can easily discriminate between the deamidated and the non-deamidated forms to assess the amount of deamidation within the sample. The critical point to mention here is that, particularly on the AcuityQDA, the requirement is that each of those species would be chromatographically resolved because of the mass resolution of the instrument. If they were perfectly co-eluting, the QDA would not be able to discriminate between deamidated and non-deamidated. So that is a prerequisite for doing this analysis. We then move on to something on the larger scale, so something very large. We often get asked, what's the largest peptide or largest species that you can analyze the QDA? And shown here is a fully reduced heavy chain glycosylated peptide. And you can see that we have a vast array of different charge states for that heavy chain, which is roughly 50 kilodalton, as well as the deconvoluted spectrum on the top, where we can easily see the glycoprofile for the heavy chain. The point being that the limitations of using the QDA are not related to the size of the molecule, but it's more related to the ability of that molecule to adopt multiply charged species, which would fit within the QDA mass range. We'll look at another example, which is using hides, which essentially takes the monoclonal antibody and chops it up into 3 equivalently sized components. Again, we see that we're getting very good mass accuracy as well as very clean spectra for each of those components, and the deconvoluted spectra match very well with what would be expected for that analyte. So I want to transition now and discuss a case study. So this is a collaboration we had done with one of our customers, which is a large biopharmaceutical developer and manufacturer, and they were interested in how the QDA would fit their needs for starting to implement multi-attribute methods. So they supplied us with a range of different samples, and then they wanted us to generate results in our laboratory, and then they compared our results to the results they collected in their own laboratory on their orthogonal QTOF instrument. So in the following slides, I'll give you a snapshot of how the QDA performed in each of these analyses. So the first give you a comparison of a peptide map, and this is looking at a comparison between looking at the TUV trace versus QDA trace. You can see here, if we look at the first 30 minutes of this lyc peptide map for each of the components within the TUV, we have a corresponding signal within the QDA. Similarly, as we move into the latter part of the gradient at 30 to 60 minutes, each of the peaks has a partner. Again, 60 to 90 minutes between the TUV and the QDA, there's very good agreement. And then finally, as we get to the end of the separation, looking at the 90 to 120 minute, good correlation between the optical and the QDA. To expand on that, we took the same samples and we did an analysis to ensure that we weren't missing any components within our analysis. So we did in our own lab an analysis on our Synapse platform and then compared that to our QDA. And as I show this, the point here is not to say that the QDA can replace the Synapse instrument. Obviously, a Synapse is required, or a higher-end MS instrument is required to characterize the molecule of interest. But for routine monitoring, once the identity of each of those peaks is known, can the QDA detect and monitor each of those peaks? So shown here is the 0 to 30-minute trace. And the chromatograms are offset somewhat, and that's due to the fact that they were each run on a different chromatographic platform, Synapse on an I class, and QDA data on an H class. Look at the 30 to 60-minute window. Again, there's very good correlation between the Synapse data and the QDA data, with each of the species being present in both. In the 60 to 90-minute run, again, a similar profile between two different instruments. And then finally, at the end of the gradient, again, the same agreement between the two instruments. What's interesting here is that in this lyc digest, the peak highlighted, or with the red arrow pointing to it, that's the raw spectrum for that conserved peptide that we had seen earlier, which is the conserved peptide from monoclonal antibody. If we take that spectrum and then compare it to the data we had seen earlier, which was from a triptych digest, and compare that to the lyc digest, these are two separate digests done in two different labs, run on separate days on different instruments. And we see this very good agreement both in the intensity as well as the profile for the charge states for that peptide across those two different experiments. So it highlights the robustness of using this instrument and looking at the same type of analyte over multiple days from different labs. We'll look at more closely at some of the other results we obtained during this collaboration. First, looking at relative quantification of native versus oxidized peptides. We show here that with the QDA, we get very close results to what the customer had attained in their own lab on their own instrument, the orthogonal instrument, for relative quantification of the oxidized species versus the native species. So, very good agreement between those two experiments. Then look at the deamidated species. So in this case, we have a peptide from the heavy chain, and then comparing that to its deamidated form. We get very good agreement, again, between the QDA as well as the instrument in their laboratory, highlighting the general utility of the QDA for monitoring some of these critical quality attributes as part of a multi-attribute methodology. Another example of looking at deamidated species, in this case, highlighting the use of two separate selected ion chromatograms or selected ion recording for each of those, and then overlaying them in sort of this view. So they look like one chromatogram. Again, we get very good agreement between the results required on the QDA compared to an orthogonal TOF instrument between those two experiments. And all of this, again, all of these results I've shown so far were collected within a single chromatographic run. A question that we often get asked is the applicability for glycopeptides, which certainly is of interest, not only just the glycopeptides, but also the glycans themselves, which we'll discuss in a few moments. If we look at the glycopeptides, in this case the G0F, G1F, G2F peptides within this peptide map, again, we get very good agreement between the results that we obtain on QDA as well as on the orthogonal instrument collected in the customer lab. So overall, looking at all of these results, this highlights the general utility of the QDA for doing routine monitoring of peptides, particularly as molecules would move downstream, and the desire would be to move those on to more easy to implement mass spec platforms, of which the QDA would be an ideal candidate. So with that, I want to transition now and spend some time in discussing the application of the AcuityQDA for routine and glycan monitoring. So in this section, I'm going to discuss the application of the AcuityQDA for routine glycan monitoring. And I want to draw your attention, as I will at the end, to the fact that a lot of the background information about some of the technologies that I'll discuss here can be found in some webinars that we've delivered earlier this year. And you can listen to and learn more about the specific technologies that enable the use of the QDA for doing release glycan analysis. So to begin, I want to introduce the use or the new reagent, RapiFluor-MS, a reagent that we introduced earlier this year at WCBP in Washington, D.C. And the introduction of this labeling technology really is built off of our existing expertise in doing rapid labeling coupled with fluorescence that we introduced in our AcuFluor reagent, which is used for doing amino acid analysis. In the creation of the RapiFluor-MS reagent, we took that information, which is the rapid labeling piece, which is highlighted here under the Rapi, in the Rapi bubble. Coupled to that, a quinolol fluorescent tag, which gives us much improved fluorescence intensity compared to traditional labels, and really gives us a boost in the fluorescence intensity. And then the additional modification was the introduction of a tertiary amine tag, which is shown on the right. And that gives us the added MS sensitivity necessary to apply a technology like the QDA for doing release glycan analysis, particularly using an MS instrument like the QDA. And as I mentioned, more information on the label and its use can be found in other webinars, also available from Jen. So if we look at some of the benefits of using the RAPIFLOR-MS label for glycan analysis, if you're familiar with a conventional workflow for doing sample preparation for glycan analysis, a conventional approach can take anywhere from hours to, in many cases, we hear multiple days to do a full release labeling and analysis of a released glycan sample. What we've done with the RAPIFLOR-MS is not only introduced a new labeling reagent for doing that analysis, but also introduced an entire workflow that spans from the release of the end glycans from the protein in a partnership with New England Biolabs, coming up with a rapid PNGSF release strategy, a very quick labeling reaction, which can take, the procedure takes only 5 minutes, and that's owed to a large part to the reactivity of the labeling reagent itself. And then a single purification step at the end to purify all of the byproducts from the labeling step as well as the excess protein generated after PNGSF release of glycans from protein. So overall, we've reduced the total sample preparation time down to 30 minutes by the introduction of the GlycoWorks RAPIFLOR-MS and glycan kit. If we look at the application of the RAPIFLOR-MS labeled glycans and look at the relative responses in both fluorescence and MS compared to some conventional labels. So we can see on the left and the right-hand side of the slide, we have a comparison in both the fluorescence as well as the MS, looking at the base peak intensity, average base peak intensity for the species across the separation. So if we compare first to RAPIFLOR versus Instant AB, you can see roughly a 2-fold increase in fluorescence and a 1,000-fold increase in the mass spec response of RAPIFLOR-MS versus Instant AB. And it's worth pointing out at this point that the comparison in fluorescence, we've taken the literature-reported maximum excitation and emission wavelength for each of the labels and used those for comparison. We get the most accurate, fair comparison of each of the labels. We then compare RAPIFLOR-MS against a more traditional label, which would be 2AB. We get roughly a 14-fold increase in the fluorescence signal and a 150-fold increase in the MS response. Finally, if we compare RAPIFLOR-MS against procainamide, again, we get a corresponding 14x increase in the fluorescence signal and a 3-fold increase in the MS signal. So compared to all the traditional reagents on the market, the RAPIFLOR-MS offers a benefit both in fluorescence. So if we look at the implications of the comparison, particularly focusing on the MS improvement, what we show here are 2 different glycan structures, which are fairly common within monoclonal antibody. We show representative spectra for both a RAPIFLOR-MS as well as a 2AB labeled glycan. Highlighted in the green box is the mass range for the AcuityQDA. So what you can see by using a traditional label shown on the right-hand side, for each of those structures, there's no charge state that actually fits within the mass range of QDA. And in addition, the MS response is 1 to 2 orders of magnitude lower than we would see compared to RAPIFLOR-MS. So if we look at the RAPIFLOR-MS side, we see not only do we have charge states that fit well within the mass range of the QDA, but in addition, the signal to noise and the relative intensity of each of those structures is sufficient to enable us to monitor each of those structures. So we look at how this can be applied for doing a routine end glycan analysis. In this case, looking at 3 different samples prepared separately. First being an IgG glycan pool, which represents really the simple biantennary structures, probably the most common structures encountered within the biopharmaceutical market today. In addition, RNase-B, which represents the high nanostructures, which are often monitored as critical quality attributes and also stability markers for a process in manufacturing the biopharmaceutical. And finally, in looking at something complicated like bovine fetuin, which represents the large, complex, and highly silated structures. What we see here is on both the top and the bottom, which are fluorescence and MS traces, there's very good correlation. So each of the peaks in each of the chromatograms is present, meaning regardless of the type of detection used, you can see each of the structures. And I purposely left the labels off of each of the traces because of the high similarity between those two different analyses. So if we look at them again now with the label, the fluorescence trace on the top and the total ion chromatogram on the bottom, we see there's very good correlation between those two different detection modes. And that allows us then to not only get quantitative information by using the fluorescence trace, but also to get quantitative information within the same experiment by monitoring with the QDA as an inline detector. So all the flow that goes through the fluorescence will go into the QDA, and we get both data streams from one analysis. A common question that we'll encounter when we start discussing this work is, what's the sensitivity level to which you can achieve with the QDA for doing glycan analysis? So in this case, we have on the top our fluorescence trace, and the bottom the total ion chromatogram from doing the QDA analysis. And on the right-hand side are representative spectra for both the most intense as well as one of the lower intensity structures with separation of IgG-related glycan. So whether we have structures that are at the high end of the relative peak area based on fluorescence or at the low end, we're getting very good spectra, and we can certainly go lower than 0.5% relative peak area. There are some examples we have go down to 0.1 and sometimes below 0.1% relative peak area. We still have very good signal-to-noise and high-quality spectra. So if we look at something more complex, and this is a separation that we had seen earlier, which is the separation of bovine petulant released glycans labeled with Rapiflora-MS. Again, we have very good correlation. And as I mentioned, these can be very complex, very highly dilated structures. So if we look at the spectrum for one of those structures, in this case we're looking at a triantennary tetrasilated structure, and we get very good MS response for that structure, and the mass information here is enough to allow us to monitor and confirm the presence of that structure within that separation. As I mentioned earlier in the presentation, the QDA integrates very well within Empower, and what that allows us to do is enable the technology to be used in a more routine analysis. So in this case, we have an example of a set of glycans. These are actually represent glycans related to IgG. This is actually a separation of a standard that we sell, the Rapiflora-MS labeled glycan standard. And for each of these components, we've assigned a component name, which is the basic name shown above each of the peaks. And the other value above each of those peaks is the base peak mass for each of the components of that separation. And all of that information is easily annotated within Empower. So not only do you get the annotation, but then you also have the reporting capabilities where you could report all of this information to be able to make decisions and have information about your analysis. And shown here is an example of a report. While it's admittedly somewhat difficult to read, we have an example of the chromatogram as well as a tabulated list of all structures within that separation. And shown on the right-hand page would be a comparison of different analytical runs, or could be different batches. And we can look very easily at the structures across different separations and see if there are any structures that are maybe increasing or decreasing in intensity. An additional application that I wanted to discuss, this has been an application that we had some thoughts on, and after we'd done the work and presented this to a number of our customers or potential customers, it's gained a lot of interest. And that is in developing a very rapid method for doing released N-glycan analysis. So in this case, we've taken the N-glycans that were released from trastuzumab, and we did an analysis with our normal 55-minute chromatographic method, which is a more highly resolving method. And we scaled that method down to be a 10-minute method. So the 10-minute injection to injection is a 5-minute gradient, and then including equilibration after. And what you can see between those two analyses is that we do lose some resolution between the different structures. The benefit is that we're able to get a very high throughput analysis. So if we couple the 10-minute method along with a 30-minute sample preparation on doing the RAPIFluor-MS release and labeling of the glycan structures, this represents a total 40-minute workflow to be able to start to get to this type of information. We then incorporate the use of the AcuityQDA and incorporate it into this analysis. Even though we've lost resolution according to the fluorescence, our ability to do selected ion recording for each of the structures within that sample allows us to quickly assess and do relative quantification for each of the structures that would be of interest, say in terms of process optimization or potentially in a clone selection type of assay, where you'd be looking for specific structures and you really need a quick turnaround in order to get an answer and be able to make a decision moving forward. So again, each of those structures we can clearly see and are cleanly depicted within their selected ion chromatogram. We then apply that, in this case, looking at one particular structure which is often of interest, which would be the MAN5. The first thing we see is that the separation itself is highly reproducible. So shown on the top chromatogram is an overlay of 6 different chromatograms at different levels of mannose-5 within the sample. Additionally, we see that the response on the QDA is highly linear. So as the amount of that glycan structure increases, the response on the QDA also increases. By using the QDA, we get unambiguous quantification of that structure since we're only selected to look at it. So if we apply that to then a relative quantification experiment, and in this case we have a mannose-5 that we've spiked into a sample of glycans, and then compare that to a structure which is not changing in intensity, or at least the structure that we want to have relative quantification against, we can see that at both low, medium, and high levels, we can easily do relative quantification between those two structures. And what's notable about this is in this case, both these structures are almost perfectly co-eluting within the separation, meaning without the use of something orthogonal like a mass detector, we would not be able to make this assessment in this time frame. So not only do we see this, but we've seen a number of customers respond very well to this as a potential application, particularly within process development. So with that, I want to transition now away from peptides and glycans and give you some examples of different applications that we've seen the QDA be applied for. Primarily, I want to focus on the QDA application for doing detergent analysis and then briefly on some oligonucleotide application work that we've done recently. So if we start first with the detergent analysis, I want to show you first the instrument configuration for doing this analysis. In the work here, we adopted from the literature reported by Harris and his coworkers. So we have here an Acuity system where we do our entire separation and use a column manager instead of just a regular column meter. And that gives us the ability to use the valves within that column manager to do something quite interesting. And then in this case, we do both ELSD and QDA detection. And that allows us to use the ELSD for doing quantification of the amount of detergent and then use the QDA to get a mass profile to assess the quality or even the stability of the detergent within the formulation. The benefit of incorporating the column manager within this workflow is it allows us to introduce samples that maybe have high protein content and divert that protein first to waste, and then use the valve then after the protein has been flushed from the analysis to flip the valve to put both the detectors in line for doing our quantification as well as getting the quality information on the detergent. So if we look at an example analysis, first looking at the ELSD response, we see that it's very linear across a wide range of different detergents. This is our calibration curve. We're getting very good calibration and very good overlay and reproducibility for doing this analysis. What's important then is with the incorporation of the QDA for this analysis, both at the low end and the high end of that calibration curve, and this curve represents a slightly wider range than was originally reported within the Harris paper, so well within what would be expected within a biopharmaceutical environment. We see that the spectra obtained at each of those mass load levels is very clean and allows us to assess the heterogeneity of that detergent within that formulation. In addition, we've seen the application of the QDA for looking at the raw materials, and then in formulations, not only in the final formulation, but also in extended stability testing, where the heterogeneity of the polysorbate may change over time. We then look at different types of detergents which would be commonly used within the biopharmaceutical environment. We see not only is the QDA applicable for doing something like polysorbate, which is perhaps the most common detergent used, but also looking at something like Triton X100 or Bridge type of surfactants. We see that the QDA is broadly applicable for a variety of different non-ionic surfactants and allows us to easily assess the heterogeneity of those samples. Finally, if we look at an application, in this case, this is oligonucleotide analysis, which is a growing area of interest for a wide number of customers. You see here, this is a separation. In this case, this is an oligothymidine ladder. So it's just a series of thymidines ranging from, looking at the chromatogram down below, the first main peak is a 15T oligo ladder, and then the other large peaks are increments of 5. So you go from 15 to 20, 25, 30, and then 35T ladder. The spectra on the top we collected or generated using the mass analysis window within Empower. So these represent the spectra at the apex of each of those main peaks. You see we get very high quality spectra for each of those oligonucleotides, and that allows to determine the molecular weight that's contained within that peak. And it's worth mentioning that the 35-mer oligothymidine has a molecular weight of roughly 10,000. So even that fits well within the mass range of the QDA, similar to what we have seen with the protein subunits earlier. Another interesting thing to point out is the spectrum that's highlighted in the black box. That's actually one of the impurities from the 20-mer oligonucleotide. That represents the spectrum for that 19-mer oligonucleotide, which alludes just previous. Again, we see a variety of charge states for that oligonucleotide, and all within good agreement of what we would expect. And the other note is that the peak is present at roughly a 0.5% relative peak area within the total chromatogram. We have very good sensitivity to see the structures at a low level of concentration. So in summary, during the discussion today, I hope I've shown you that the AcuityQDA really can enable multi-attribute monitoring, whether it be in doing a protein assay, a glycan assay, or other types of assays. Particularly in relation to peptides, it allows you to do the identity, assess oxidation, deamidation, glycoproteins, all within a single experiment. And that work can also be expanded to looking at the subunits, whether they be from a heavy chain, light chain experiment, or alternatively from doing something like an IDES digest or some other limited proteolytic digest. The QDA can be easily integrated into existing workflows, primarily because of its ability to fit within Empower and also the application for both doing TFA and formic acid-based separation. And what we've seen is that the adoption barrier is quite low, not only due to the low cost of the instrument, but also how easy it is to use and ease of maintenance and setup of the instrument. In relation to using the QDA for release and glycan monitoring and doing that more routinely, really the synergy of using the AcuityQDA along with RapiFlow-MS enables the use of the QDA for those types of routine-based assays. And what it brings is the ability to do both rapid sample preparation as well as whether they be highly resolving methods or really rapid separation to use the QDA to assess the structures within that released and glycan pool. Finally, I want to conclude with some additional information. So if you would like some more information on the AcuityQDA and its application in the biopharmaceutical environment, you can visit waters.com/biopharmQDA. Additionally, as I mentioned earlier, we have 2 previous webinars this year, both primarily focused on the use of the RapiFloR-MS, whether information on RapiFloR-MS for released and glycan analysis or its application with the AcuityQDA. You can visit view either of those 2 webinars, which are available now. Finally, I want to thank my coworkers, and I welcome any of the questions that you might have. Thank you very much.
Thanks, Sean. That was an excellent presentation. I think you provided our audience with some great information about how to utilize multiple attribute monitoring to increase analysis efficiency, and showed us all the sensitivity and versatility of the AcuityQDA detector. So we thank you for that. Okay, before we start our Q&A session, I want to remind everyone that this is your final chance to submit your questions for Sean, so hurry up and submit them now. So since I see a few questions already starting to roll in, why don't we get started, and we'll try to answer as many questions as we can.
Okay, Sean, so let's get to our first round of questions here. Our audience member would like to know, can QDA be controlled by mass links so both data collection and processing can be streamlined?
Yes, so the QDA can be controlled as well as in power, as I discussed, but also in mass links. So the answer is yes. The instrument can be controlled in both of those chromatographic data systems.
Okay, great. So we have another question that would like to know, what is the maximum mass range of QDA with the max range of detection? What scan frequency in hertz will it be?
So the answer to the first part of the question, the maximum mass range of the AcuityQDA is up to 1250 m over z, and that starts at 30 m over z. So that's the actual mass range of the quadrupole. In terms of the range of detection and the scan frequencies, what they will be, it will depend on the number of functions that the user is acquiring for any given assay. So the instrument is capable up to 20 hertz acquisition, but the frequency of that acquisition will change depending on the number of functions, and that capability is calculated as part of the method. So you can define a target frequency, and then as part of the inlet method, the instrument will tell you what its actual sampling frequency will be. So I can't give a concrete answer because it really depends on what you're doing.
Okay, great. So our next question comes from another audience member who would like to know or asks, can you describe the performance options available for the QDA, specifically in terms of the pumping options?
So the QDA is available in 2 different pumping options, and they're referred to in either standard performance or a high performance at a basic level. And the difference between them is the type of vacuum that is used. So in the more standard performance, it's a diaphragm pump that sits on the back of the instrument. What we found for a lot of biopharmaceutical applications is the enhanced performance, which is a very small but external rotary vein pump, seems to be the most appropriate for doing biopharmaceutical applications. And the difference in sensitivity between those two is probably roughly a factor of 4 or so. 4 or 5 fold improvement in sensitivity, and if necessary, switching between those 2 different options is a fairly simple thing to do.
All right, great. The next question is, audience member wants to know, is the deconvolution software for the QDA data available for both Empower and mass links, or just mass links?
So that's a good question. So currently, the deconvolution software is only part of the MassLink solution. It has not currently been implemented within the Empower workflow.
Okay. So the next question asks, does D-N glycosylation go to completion?
So again, another very good question. So the answer to that question is yes. We've done a a variety of different assays, gel shift assays, as well as other approaches, including MS, to look at the molecule after deglycosylation. And each of those studies, what we found is the deglycosylation goes to completion. I would mention as well that if there's a desire to get more information about the N-glycan sample prep and how that works, we've recorded earlier this year several different webinars which describe that, which are both available from Jen. And they go into a lot more detail and describe the different studies that we've done to ensure that we get complete deglycosylation, that the labeling itself is complete, and also investigated the impacts of SPE on the sample prep. And I would suggest reviewing those and viewing those to get more answers and detailed answers about specifically about N-glycan sample prep with RapiFluor MS.
All right, thank you. So this next question is a little bit long and in a couple parts, but audience member wants to know, is there any reagent kit for increasing the charge state of large peptides and proteins to make the fit for QDA detection? Does RapiFluor and the workflow have to be used with QDA, and what is its performance on other mass specs?
All right, so that is a number of questions. So in terms of a reagent kit for increasing the charge state of large peptides or proteins, currently we do not have a reagent kit for doing that. What we found for the majority of our applications is that for the most part, for most peptides, I think every peptide at this point that we've investigated, that peptide can adopt a charge state which will fit within the AcuityQDA's mass window. In particular, if I refer back to the heavy chain, light chain data that we showed earlier, one of the requirements to obtain mass information for, in particular, the heavy chain is that the peptide or that heavy chain be fully denatured, and that's to allow it to adopt enough charge. In terms of RapiFluor, does it have to be used with the QDA? Absolutely not. The benefits of using the RapiFluor MS span across all of the different fluorescence or MS detection options that are available. And that ties very well into the last part of the question, which is the performance of the reagent on other MS instruments. So we've used RapiFluor MS not only for the routine monitoring, which is what I discussed today, but also in the characterization workflows. And what we've seen is that in the characterization workflow, not only do we have dramatically improved MS sensitivity for doing full scan MS, but that the improvement in sensitivity also carries through to doing the MS-MS analysis and doing more detailed structural characterization of the glycan structures. So I guess the general answer is that it does not require you to use a QDA, but it opens the opportunity to use mass detection downstream of the more higher-end characterization pieces in which RapiFluor MS brings benefit as well.
Okay, great. Thanks for that answer. One of our other questions is, how accurate is CDR peptide monitoring?
So the accuracy on the CDR peptide monitoring, I wouldn't narrow it down to specifically the CDR peptides, but the mass accuracy of the QDA, the specification is plus or minus 0.2 Dalton, and that's at the M/Z range. And that's the standard performance, and that's generally the performance we see for each of the peptide applications that we've done.
All right, and it looks like we have time for just one more question. Our audience member would like to know, if the QDA can do what's similar to the G2, why would a customer buy an instrument like the G2?
Okay, so that probably refers back to the comparative data that I had shown, the QDA versus the SYNAPT data. So to clarify that discussion, the point is not that the QDA could replace something like a higher-end MS or QTOF or a G2 instrument. Those higher-end instruments are still very necessary in order to do the characterization to identify what the specific species of interest, whether they be peptides or glycans, are, to really understand and characterize what all the structures within the sample are. Then being able to migrate those assays to downstream more routine assays that could be done with a QDA is what has been a desire. So the point in showing that comparative data was to show and demonstrate that the structures and the signals that you see on a higher end instrument can also be observed on a QDA. And the difference between those two experiments really isn't the mass load on the column. So the QDA will require a slightly higher mass load. And it's generally in exactly the same realm as you would inject onto a chromatographic column as you would use for an optical-based assay. So it wouldn't necessarily require a change in the current protocols, just simply adding a mass detection.
All right, great. And with that, we've come to the end of our webinar. So I want to remind everyone that the webinar will be archived for 6 months on our website at www.genengnews.com. So if you missed parts of it, you can watch it again, or feel free to forward the link to your friends and colleagues, which we always recommend. I'd like to thank Sean again for his informative presentation, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to Waters for sponsoring this webinar. So hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now.